Translational medicine is where lab discoveries meet clinical reality, and it is notoriously difficult. You take an underspecified research question, like figuring out the optimal second payload for a topoisomerase I inhibitor in non-small cell lung cancer, and you have to synthesize literature, clinical trials, patents, and genome-scale multiomics data. You need to preserve exact identifiers, track uncertainty, and produce an auditable dossier that a pharma team can actually use. General-purpose foundation models and off-the-shelf multi-agent systems just aren't built for this. They tend to spit out single-shot answers, run in open-ended loops, or lose track of provenance. Today, we're breaking down BIORESEARCHER, a scenario-guided multi-agent architecture designed specifically to solve this exact problem. The core challenge here is operational. First, entity ambiguity. Topoisomerase I, and top1 all point to the same protein, but different databases index them differently. Second, evidence is scattered across PubMed, clinical trials, Gov, Google Patents, and DepMap multiomics datasets, each with distinct access patterns and biological contexts. Third, reasoning has to span interacting biological scales, requiring both qualitative synthesis and quantitative computation. And fourth, the output can't just be a summary. It has to be a ranked hypothesis list with mechanistic rationale, fully traceable back to its sources. BIORESEARCHER tackles this by separating method selection, evidence acquisition, and reconciliation. A master orchestrator takes your natural language query, maps it to a versioned research playbook, decomposes the task, and delegates to specialized, state-isolated subagents. These subagents publish provenance artifacts to a shared evidence bus. Finally, a reconciliation agent drafts, compares, and resolves claims before a light editorial pass produces the final report. Let's get into the technical stack. Everything starts with a translation subagent. It resolves entity ambiguity using a RAACT agent backed by local SQLite databases for ChML36, open targets, and PubChem, with API fallbacks for latency. It maps natural language mentions to canonical identifiers, like turning topoi into ChML target ChEMBL 1781, and ensemble ENSG 000001989000. This single grounding step prevents downstream agents from searching the same target under different names. From there, the system integrates over 30 tools across 6 major categories. You've got literature search via PubMed and NCBI utilities, clinical trial retrieval through the clinical trials, Gov v2 API, patent search using SERP API with support for both text and chemical structure queries, and target profiling pulled from DepMap and the Human Protein Atlas. For conference abstracts and license data, it uses a custom RAG pipeline with PyMu PDF chunking, sentence transformer embeddings, and ChromaDB for metadata-filtered vector search. This is crucial because conference data often reports clinical results 6 to 12 months before peer-reviewed publication. Where BIORESEARCHER really separates itself is in quantitative analysis. Instead of relying on a fixed set of pre-enumerated tool functions, it exposes a Kodak sandbox for genome-scale computations. The Insights and Signals subagent runs a sandboxed Python environment preloaded with 9 DepMap 24Q4 Parquet datasets, covering roughly 19,000 genes across 1,600 cell lines. This includes CRISPR dependency screens, gene expression, copy number variations, proteomics, and somatic mutations. The namespace comes preloaded with pandas, numpy, and scipy, plus a dictionary of expert-curated biological thresholds. For example, a CRISPR gene effect score below -0.5 flags likely dependency, while copy number gain is set above 1.5. This grounds the LLM's statistical analyses in accepted DepMap conventions and prevents arbitrary cutoff selection. Security is tightly controlled. The sandbox blocks dangerous built-ins like eval and open, enforces a 600-second execution timeout, caps output at 8,000 characters, and restricts variable persistence to MSGPAC serializable primitives. If the code hits a timeout or tries to access the network, the LLM gets a diagnostic message and self-corrects. This allows for 18 callable tool functions spanning genome-wide dependency correlations, mutation stratification using Mann-Whitney U-tests, synthetic lethality analysis, and drug combination synergy prediction. Once multiple subagents and models produce drafts, you run into a classic multi-agent problem, conflicting evidence. BIORESEARCHER handles this with claim-level multi-model reconciliation. The reconciliation agent extracts atomic claims from each draft, complete with source quotes and confidence scores. It then clusters these claims across drafts into topic groups labeled as full agreement, partial agreement, or conflict. The system then runs a multi-round argumentation loop. In parallel, each model issues an updated stance per group, conditioned on the other agent's prior positions. It continues until no model disagrees on any group, and the mean confidence crosses a predefined threshold. This structured debate ensures that the final report doesn't just average out disagreements, but actively resolves them before the editorial pass. To test this, the researchers designed a 3-tier evaluation protocol. First, single-step tests across 109 questions to verify each system layer in isolation. Layer 1 covers ontology and entity resolution. Layer 2 tests qualitative retrieval, and Layer 3 checks quantitative computation. BIORESEARCHER, powered by GPT-5.5, achieved an 83.5% pass rate, and a 0.892 average judge score, outperforming specialized agents like CellType and frontier models like GPT-5.5 and Gemini 3.1 Pro. The biggest margin was in quantitative analysis, where it hit over 91% compared to under 30% for the baselines. Second, they tested autonomous analysis on Bix Bench and BI's Bench Scientific Discovery. BIORESEARCHER scored 89.33% accuracy on the human biology subset of Bix Bench and matched Claude code on the BAISSD track with a mean score of 0.758. Third, and most importantly for translational medicine, they built a 30-query end-to-end clinical benchmark. Each query pairs a drug or mechanism of action with a clinically validated biomarker, plus a carefully crafted negative control from adjacent biology to test discrimination, not just recall. BIORESEARCHER achieved a 74.7% positive hit rate and a 96.8% negative clear rate, leading all baselines on both metrics. Let's walk through a real case study to see this in action. The team tasked the system with generating synthetic lethality biomarker hypotheses for ATR inhibition, based on the TRESR clinical trial. The orchestrator routed the query to a combination discovery playbook. The translation subagent canonicalized ATR and the relevant indications. Then, the CODACT subagent autonomously selected TCGA and METAPRISM patient cohorts, identified loss-of-function events associated with ATR expression, and ran survival analyses across multiple time points. The system integrated patient-level data with literature and clinical context, evaluating two signal types, loss-of-function-associated ATR upregulation and survival patterns in low ATR tumors as a proxy for drug-induced inhibition. It ranked TP53 loss as the strongest pan-cancer marker of ATR pathway activation, ATM loss as the most clinically supported synthetic lethal candidate, APC loss in colorectal cancer as an unexpected data-driven discovery, and ARID1A loss as biologically plausible but weaker in the available patient data. Crucially, the system showed calibrated uncertainty handling, explicitly downweighting unstable Cox interaction models and deprioritizing inconsistent signals like CDKN2A. The entire pipeline ran autonomously in roughly 22 minutes, producing a fully traceable translational dossier. BIORESEARCHER demonstrates that orchestration alone doesn't solve autonomous biological discovery, but when you combine it with entity grounding, scenario-specific methodology, source separation, and claim-level reconciliation, you get a system that retains competitive data analysis ability while providing the exact machinery scientific users need. It's a major step toward auditable, reproducible, and clinically actionable AI-assisted drug discovery. If you want to dive into the architecture details, benchmark construction, and full code specifications, I've linked the paper in the description. Thanks for watching, and I'll see you in the next one.